And so our final speaker for this session is Ahmed El-Sohemy, and he is going to present his work both from the research and practical experience, as well as addressing some of the questions surrounding genetic testing. And he's been recognized for numerous awards and is the founder of Nutrigenomix, Incorporated. Ahmed?
Thanks very much, Naomi, and good morning, everyone. It's a great pleasure to be here this morning, and like Jose, I also remember the first workshop that was held here about a decade ago. It was in a larger auditorium, and at the time I attended just as an interested junior young faculty researcher doing work in this field. So it's a great honor to be invited this time to actually present to you some of the work that we have been doing in this field, and just touch on some of the translational activities through our startup company. So in addition to my day job at the University of Toronto, I'm also the founder and serve as CSO for the company Nutrigenomix with an X, which is not a direct-to-consumer genetic testing company. It's a service that we provide only to healthcare practitioners, mostly registered dietitians, some physicians that practice functional and integrative medicine. So we currently have just over 6,000 practitioners in 35 countries and reports available in eight different languages. So I've entitled my talk as Genetic Testing for Personalized Nutrition Ready for Prime Time. And spoiler alert, my answer is yes. I agree with some of the comments that Dr. Alpers made in the previous presentation, but what you'll see is I'll present evidence to show that many of those criticisms, which I often hear, are actually not true. And so I'll go over some of those points. And just by way of disclosure, I am, as I mentioned, the founder, and I do hold shares in Nutrigenomix. So the field, as we all know, is not without controversy. There are obviously many kinds of operators out there, and I think we've heard from some of the previous speakers that the evidence base, the science base behind many of the different offerings out there is quite varied. And some is certainly what you can call perhaps on the fringe or not really rooted in robust scientific evidence. And so we see all kinds of these articles posted that question the validity behind these kinds of tests. And I think it is important to keep these companies honest, but I think it's also not right to lump all of them in one basket and say this whole field is all just snake oil. Part of the controversy, I think, comes from the different kinds of tests that are on the market. And just focusing on the genetic piece for the moment, I think it's important to make a distinction between disease risk genes that are often identified through genetic association studies versus what I like to refer to as the modifier genes or the metabolic genes. So these are the genes where alone are not related to any phenotype or health outcome, but they modify the effect of an environmental factor. So think of a drug metabolizing enzyme or a drug transporter. Having a genetic variant, for example, doesn't necessarily cause any adverse effects, but if you have a particular genetic variant and you are prescribed a certain drug, it may cause adverse drug reactions. So that is the analogy that we take from the field of pharmacogenetics, and we know the same applies to nutrition as well. So why is it important to look at genetics in the field of nutrition? I'm sure all of you here in the room can appreciate that if we look at the link between virtually any nutritional factor and any health outcome, if there have been enough observational studies, we see this heterogeneity of response. Nathan showed a nice slide showing all those associations between the different foods and cancer risk. Some studies show increased effect, some no effect, and others the complete opposite effect. Now, there are a lot of reasons for the inconsistencies between these studies, but an important consideration, of course, is the genetics of the group or the population that you are studying. And so I've just tried to illustrate kind of in a black box potential genotypes that can help us predict whether or not a nutritional factor will increase the health outcome, have no effect, or decrease it. So again, you think of something like sodium and blood pressure. Some people that go on a low sodium diet, their blood pressure goes up. Right? We used to think of them as outliers, try to figure out how to get rid of them from the statistical analysis. But more and more, these so-called outliers are there, and they're very real. And so what if you are an outlier, and the advice that we are giving you is actually causing harm? And if there's a way of identifying these individuals, finding alternate strategies, that's obviously something that we need to consider. So obviously, one size does not fit all. So Jose had mentioned a nice example of ApoA2 saturated fat, and he used that as a proof of concept. So what I wanted to do, just because the interest of time, can't really go through a comprehensive review of all the scientific evidence, is just to use one example here as a proof of concept. And this is some of our earlier work looking at caffeine and cardiovascular disease. And we reported this over a decade ago in JAMA, where we found that the fast metabolizers, those on the left-hand side, moderate consumption of coffee actually lowers or is associated with a lower risk of myocardial infarction, whereas the slow metabolizers on the right-hand side, even 2 to 3 cups a day, is linked with a higher risk. So if you look at the fast metabolizers, 2 to 3 protective, the slow metabolizers, the complete opposite effect. So this was back in 2006. A couple of years later, this group from Italy essentially replicated our findings, but rather than looking at myocardial infarction, they looked at the risk of developing hypertension. And so this was a prospective study. So they recruited a bunch of pre-hypertensives, they genotyped them, assessed their coffee consumption, and then monitored them over a few years. And they found a similar pattern. So in Italy, cup sizes are a little bit different, but nevertheless, you see the same pattern. On the left-hand side, fast metabolizers are protected, slow metabolizers, complete opposite effect. A few years later, similar phenomenon with prediabetes and impaired glucose tolerance. Slow metabolizers, increasing coffee, increases the risk of impaired fasting glucose, no effect with the fast metabolizers. We collaborated with this group to look at kidney function. As you can see on the y-axis, glomerular filtration rate. So usually a high GFR is good, but if it's too high, it's indicative of impaired kidney function. Your kidneys are overworking. And you can see here the regression line with increasing cups of coffee. The fast metabolizers, no increased risk, but the slow metabolizers, a strong increased risk. Other work that we've done, so these are all observational studies, but we've also done an intervention trial, but looking at endurance performance in athletes. This is something that we can obviously do in a short-term trial. And so we conducted a randomized controlled clinical trial where we recruited individuals and based on genotype, randomized them to receive placebo or 2 different doses of caffeine. And this is unpublished data, and I don't have a slide for it. But what we found was those who are the fast metabolizers actually benefit more, and the slow metabolizers, absolutely no improvement in performance. So in terms of health outcomes, you have myocardial infarction, hypertension, prediabetes, and kidney function, all showing the same thing, that if you're a slow metabolizer, you should probably limit your intake to no more than 2 cups a day. If you're a fast metabolizer, you're lucky. You can follow the general recommendations, which suggest that you can drink up to 4 cups a day. So figured I was coming to Washington, so I should refer to an article in the Washington Post. This was actually a couple of years ago, and after the guidelines came out, and the journalist here noted that the panel said coffee is safe to consume, but that might be true for only half of us. And so he obviously cited some of the work that refers to the slow metabolizers and the body of evidence and wondered why we're still making these one-size-fits-all recommendations when the science suggests otherwise. So one of the quotes in the article was by Dr. Greenland from UCLA, who noted there are spectacular metabolic differences in people, and to expect that coffee will have the same effects is absurd, and I agree. Interestingly, they also interviewed a member of the panel who noted that, unfortunately, because genetic testing is expensive and rarely done, most people have little idea of which gene variant they carry. Fair enough, right? There's no point in making recommendations based on a bit of information that people don't have access to. But what this tells me really is 2 things, is if genetic testing was cheap and everyone knew what they had, then the recommendations would, to some extent, take into account the science. So this really gets into now the economic and social aspects of genetic testing and making this kind of information available, which is a very legitimate topic of discussion. But in terms of the science, I think these are some really good examples of a proof of concept at how an individual SNP, single SNP, can modify the association between a dietary component and a variety of different health outcomes. Now, if you recall, just last week, this study came out in BMJ and got all kinds of media attention and headlines like this with the BBC, 3 cups of coffee can actually be beneficial. And this was a massive study. It was a review of 200 meta-analyses. So you can imagine the sample size that went behind this. And overall, the totality of evidence, it suggested a protective effect for a number of health outcomes. Well, at the end of the day, that just tells me that the majority of participants in all of those studies were probably fast metabolizers. In our study, we actually had 50-50. So you can imagine if you do this population study and 60% are fast metabolizers and 40% are slow, then that will be the effect that predominates. But at the end of the day, people want to know what they can do for themselves. And when they see headlines like this, they falsely assume that coffee is safe to consume. But if they're a slow metabolizer, following those recommendations will actually increase the risk for all those health conditions. So I think we need to move away from these kinds of studies, and bigger is not necessarily better. You have to look at the quality of the scientific evidence. So fortunately, just last summer in the New York Times, Ahmed wrote this piece, and he again reminded us that even though this research has been done several years ago, that the associations seem to be dependent on your individual genetic variation. And just last year, Rafael de Caterina and I put together this first consensus article through the International Society for Nutrigenetics and Nutrigenomix, where we tried to create, as an example, a position paper that reviews the scientific evidence that would enable us to make DNA-based dietary advice. And more recently, this paper has not yet come out, but it was recently accepted in Genes and Nutrition. So Jose, myself, and some others are co-authors here, where we try to propose certain guidelines for evaluating the scientific evidence for making DNA-based dietary advice. So again, there's no shortage of articles that really splash cold water on the field. This is the most recent one. This was just last week, November 27th. DNA-based dietary advice, big business, little scientific evidence. Unfortunately, when you look at the articles and who's often cited in these, here's just an excerpt. A pediatrician that was interviewed, I don't know him, but he's the author of The Bad Food Bible, which I haven't read, but anyhow, he says there's no evidence that some people respond better to high-fat diets while others are more receptive to diets packed with protein. He says it just doesn't exist. Well, Jose already presented some evidence looking at ApoA2 and saturated fat. And the fact is, this is just a statement that's based on a lack of knowledge of the scientific evidence. Because when you look, you see studies such as this by Lou Q's group at Harvard, the Pounds Lost study, a 2-year randomized controlled clinical trial comparing the effects of a high-protein versus low-protein diet. So there's a lot of people that advocate a high-protein diet for weight loss, things like the Atkins diet and other types of diets, because the fact is they work in some people. And it turns out it depends in part on your FTO gene variant. And you can see here, this is after 2 years. On the y-axis is change in fat mass. There was no difference in TLEO carriers. There's absolutely no difference between those who are on a higher low-protein diet. But look at those who had the AA genotype. Those who were on a high-protein diet lost a considerably greater amount of fat and maintained that loss 2 years later. So when it comes to the gold standard of scientific evidence, I think this is pretty robust. And it has since been replicated in a distinct population. This was a study in Spain that also showed that a high-protein diet is effective for weight loss only in those with the AA genotype. We also sought to replicate this, but because we have a cross-sectional study, we looked at existing body mass and waist circumference, and we did this in another ethnic group, in East Asians. And you can see here, recall the AA genotypes. These are the groups that benefit from going on a high-protein diet. And you can see when they're not on a high-protein diet, they have roughly a 10-centimeter greater waist circumference. It's about 4 or 5 inches or so. So again, replicated in distinct populations. You heard from the previous speaker about the FTO being used as a predictor for obesity, but what this shows is that it's also a modifier gene of protein. So it's important not to just look at FTO as a risk factor for obesity and tell people you have a higher risk for obesity, so eat less and exercise more. What you can tell them is specifically you're more likely to benefit from a high-protein diet, where someone else might not. They might need to focus on other macronutrients where they might be more metabolically responsive. So how do we balance the advice then that we currently give to populations? So I know Sylvia mentioned this morning that it's not an either or, and I agree. Hopefully we have some kind of integrated model. But currently, we are giving recommendations to populations, public health recommendations, and people want to know what works for them. Some argue that giving people genetic information doesn't really do anything, doesn't motivate them. We thought that's an important question. On the one hand, someone can say, I have the gene, I'm going to watch what I eat. The flip side could be this concept of genetic determinism or fatalism. It's in my genes, there's nothing I can do, leave me alone. But it really depends on the kind of genetic information, right? So if someone's a heavy coffee drinker and you tell them you're a slow metabolizer, they can cut back the coffee. Right? If you're told you have the risk variant for salt-sensitive hypertension, then you can lower your sodium, right? As opposed to just generic advice. So we decided to put this to the test and conducted a randomized controlled clinical trial comparing DNA-based dietary advice to standard recommendations. And we found that those who got the DNA-based dietary advice actually understood the recommendations to a greater extent. This was somewhat surprising, but some would argue that genetic information is too complex. People won't understand it. Well, it really depends on how you frame the messaging. Right? It's not that difficult for someone to understand, I'm a slow metabolizer. I don't get rid of caffeine efficiently. I have to limit my caffeine consumption. So they also indicated they were more motivated to change their eating habits. And we reported that they did, in fact, change their eating habits, and they maintained that 1 year later. Biggest effect we saw was with salt-sensitive hypertension in those who were told they had the risk variant for that. This has since been replicated, this concept of DNA-based dietary advice motivating behavior change by this group in Finland. They gave a different kind of piece of genetic information and looked at other dietary outcomes, but nevertheless demonstrated the proof of concept that giving people the right kind of genetic information. We're not talking about disease risk, telling them they have ApoE and a higher risk for Alzheimer's, and they don't really know what to do with that kind of information. It again has been replicated in the food for meat trial. And you heard from the previous speaker that there's other kinds of personalized information. In this particular study, the authors actually concluded that any kind of personalized advice motivates adherence to the Mediterranean diet. But if you look at the results, it's only in the group that got the genetic information. They just lumped them all together and concluded, I would argue erroneously, that any kind of personalized information motivates change. I actually wrote a letter to the editor noting that, and they said, no, we decided a priori that all these groups should be lumped together, which is false, because on their clinicaltrials.gov registration, they clearly wanted to test these different effects. So they wanted to test the effects of personalization based on diet, personalization based on diet and phenotype, and then diet and phenotype and genotype. And that's a way really to kind of try to mask the effects of genotype. But regardless, it was only in that group, when they got the genetic information, that they actually adhered to the Mediterranean diet. And again, food for meat trial showing, as you heard earlier, greater effects of genetic information. This is data from Robert Green's group at Harvard, where he looked at what happens when you give people genetic information and what they do with that. And you can see here that 42% changed various behaviors, a lot of them exercised, and the vast majority, of course, altered their diet. So does everyone change their behaviors? No, not necessarily. But it shows that this can be a very useful tool for getting some people to change their eating habits. We're always giving messages for healthy eating, and it's just going over some people's heads. And what this line of evidence shows is that for some people, a good number of people, that giving them the right kind of information can actually motivate them to actually adhere to the recommendations. And this was just the study that came out with a little bit more detail from their group. So there are a number of criticisms, and just in the interest of time, I won't go through all of them. The Academy of Nutrition and Dietetics had this position paper back in 2014, where they referred to these kinds of tests as being complex and not being based on actual risk. But we're not talking about actual risk. We've never talked about actual risk in nutrition. It's always about relative risk. Right? You can't even tell a smoker what their actual risk is of lung cancer is unless you know how long they've been smoking, how old they are, and other factors. So this is really a message to spook dietitians away from this and tell them, you don't understand this, and it doesn't really tell us anything informative. Fortunately, just a couple of months ago, they pulled their position paper, and they're actually reviewing, reanalyzing the scientific evidence, and hopefully we'll be coming out with another one next year. So just to briefly go through some of the comments that we often hear from skeptics that I think are not true. Single SNPs are useless. This often comes from genetic epidemiologists that are trying to come up with gene risk scores to predict disease without considering environment, and we know that that's just not true. There are a number of very good examples of how single SNPs modify the effects of specific dietary factors. Some argue, well, people aren't going to change their behaviors. We've shown to the contrary. And since when has that really been an issue, right? We know that there's still people who smoke, and it's not like they haven't heard that smoking causes lung cancer. It doesn't stop us from giving that message. Some argue that it's all about the microbiome, and I won't get into great detail, but there's very good evidence that the host genetics determines, to a large extent, the kinds of bacteria that colonize the gut. And then we need to integrate the omics and these other factors. And again, we can go through each of those if we had more time to indicate why these are all not necessarily valid criticisms. So where are we today? Well, we need to eat. And we currently give dietary recommendations on healthy eating, but that's based on some level of science, and I would argue old science. So we have to ask the question, how much more science do we need to say before we could actually start using this kind of information? And it's not meant to be revolutionary, it's just meant to be evolutionary. Are you at this end of the spectrum or this end of the spectrum? So I'll just leave you then with all of these points of why I think we're ready for prime time. And I will thank some of the members of our group, our funding partners, and thank you for your attention.